Thank you very much. Thank you. So I apologise to everyone. My voice is going slightly, hence the husky tones. So I'm sorry about that. I'm just going to start sharing my screen. Hopefully that will come through. Can everyone see my screen?
I think it's still loading.
No, I can't. I think I can't, Louise.
Okay, I've got the message that it's screen sharing my end. Let me just stop it and try it again. I think my computer's having issues. Let's try this again. We should all be used to this by now and not have any more problems.
Yeah, all good. We can see it now. Or at least I can see it.
See it now, yeah.
Thank you. So I'm going to talk to you today about low kV EDS, and thank you to the organisers of this for inviting me. I'm really pleased to talk to you today about it. And I've been using a low kV EDS for a few years now, and I think it's really exciting. So I'm very happy to share what I've been doing. So a little bit about me. I had a brief introduction earlier, thank you, but to give you a bit more of a background, I originally am a biologist, and I was lucky to do all my degrees at Aberystwyth University, right on the coast, as you can see here. And I did a master's in biological electron microscopy through the university, but actually got to go to Davos in Switzerland and do research there looking at implant materials. I then had 2 postdoctoral positions at UCLA in Los Angeles. So again, some lovely coastal regions. And then I moved back to the UK and I was a facility manager at Oxford Brookes University for just over 7 years. And we actually renovated the entire facility during my time there. I then made the leap and moved to commercial and moved to industry, which I have been very happy to have done. And I moved to Oxford Instruments. So if you're not familiar with Oxford Instruments as a company, we were the first proper commercial spin-out company from Oxford University. And we worked very heavily in MRI and superconducting magnets, and have expanded since then, really. And it's a really nice, successful British company story. We now have a global footprint. So we've got offices all around the world, and we've got a range of kind of business units under the Oxford Instruments umbrella. I work for nanoanalysis, and we're the unit that produces detectors and nanomanipulators that go onto electron microscopes. You may be familiar with some of these other names up here. Asylum do atomic force microscopes. VTech has recently joined us, and or do confocal microscope cameras and confocal microscopes themselves. So you may be familiar with some of these names, all under the Oxford Instruments banner. I work in High Wycombe, which is where the headquarters for nanoanalysis is, and typically this is one of the demo instruments we have there with our Ultimate Extreme detector on it and collecting some data. Now, I love microscopy. I fully embraced it when I did my master's degree, and electron microscopy has always been absolutely fascinating to me. Every time you put a new sample into the microscope, it raises more questions, it's fascinating, you get into it. And I've always enjoyed applying colour to what are traditionally grayscale electron micrographs. So here we've got a spider, and here we've got some 3-dimensional data. This is a connecting cilium in a rod photoreceptor cell. And here we've got some of the membranes that receive the light signals. And this has been coloured. This is a 3-dimensional data set from a TEM, and this has been coloured to show the intensity of electron signal. And here we've got some serial block face SEM data of Golgi bodies. And here we've used colour to differentiate between the Golgi apparatus and the endoplasmic reticulum that it's connecting to. So colour can be used in different ways. It's really helpful to facilitate our interpretation of data. And it can either be done for visualization purposes, as you can see here, or we can actually have it mean something about the sample itself, which is what we do with EDS. I really like this quote, and I often show it when I'm giving talks or seminars, and it's an old quote, but I think it's really applicable for microscopy. With a microscope, you see the surface of things, it magnifies them, but it does not show you reality. It makes things seem higher and wider, but do not suppose you are seeing things in themselves. And it's always a reminder to us that what we're seeing is an interpretation of our sample, particularly with life science as well. It's one aspect of the sample, and we may need multiple different types of information to truly understand what is actually going on. And this is a micrograph I collected as part of a collaboration with Rob Kessler, looking at air particle pollution on the surface of leaves. And these are all various different particles that have accumulated on the surface. And this is an electron micrograph where we've applied colour, and you can see down here the elements, and each of these, the signals from each of these elements is coloured differently, and you can see this blend of information. And that's really helpful to interpret visually what is going on in our sample. So for those of you who are not familiar with EDS, I wanted to give you a bit of an introduction to it, and it's energy dispersive X-ray spectrometry. And this is produced as a result of the specimen beam interaction. You can see an interaction diagram here, and we've got the electron beams, the electrons coming in from the main beam and interacting with the atom. And one of those interactions transfers energy to an orbiting electron, which then results in it being excited and emitted from the sample. And this destabilizes the atom. So electrons from outer layers will jump down into that space left by the inner layer electron. And in order to do so, they need to release energy, and that tends to be in the form of characteristic X-rays. They're characteristic because the energy difference between these layers can be measured and can be used to identify the atom from which the X-ray originated from. And you can see the layers are labeled KLM. So when we've got an electron jumping from the L layer to the K layer, that's a K X-ray. So we're able to detect not only the atoms, but the layers from which the X-ray originated from. And then what we can do is we can accumulate all these signals, the secondary electron signal, which gives you a lot of topographical information, the backscattered electron signal, which, because it's due to the main beam electrons being deflected by the atomic nuclei, where you get a higher density material, you get more of a backscattered signal. And you can see that here we've very clearly got some density differences. And then we can introduce colour, which is the elemental composition of these areas. And this is a barrier between an implant material down here and bone in this example. Now, EDS can be done in a TEM and an SEM. I'm going to be focusing on the SEM side. And what happens is X-rays are emitted as the beam scans across the sample, and they're detected with a detector that sits alongside the pole piece inside your microscope. So this is inside the chamber of the microscope. You've got the nose of the detector sitting very close to the pole piece there. The signal that is generated goes to a processor, and that processor then counts and measures the X-ray energies, and it produces a spectrum like this Along the bottom, we have the X-ray energy, and where we've got a peak, this is where we have an accumulation of X-rays at that particular energy. And because we know what the energy should be for different elements, we can then label it, as you can see here. Now, this spectrum is produced at every position the beam is as it scans across the sample. So we can then produce an elemental map and look at the chemical distribution across that entire sample, as you can see here. Now, one of the key things about specimen beam interactions is the volume. So as your beam hits your sample, you have a large volume where you get those electrons from the main beam interacting with your specimen, and you get emittance of all the different signals from all those areas that the beam interacts with, but only certain layers can leave the surface of the sample. So here, where we've got the main beam electron, we've got this area just below the surface where we get secondary electrons that can be emitted and detected. They will be emitted throughout this entire volume, but we can only detect those that leave the sample from this region. Slightly deeper on, we get a signal of backscattered electrons, and then we get the X-ray signals generated much deeper within the sample. And this is quite important because it affects the resolution of the data you get. And you may have better electron resolution than you do X-ray resolution because the X-rays are coming from much deeper within the sample. Now you can control this by looking at your accelerating voltage. And this is a Monte Carlo simulation. And this is in iron, and really showing what the trajectory of the different electrons are when they hit the sample, when they come from a 20 kV, 10 kV, 5, 3, or 1. And you can see the volume massively decreases as you go down in kV. Now, low kV for biological samples has been used for quite a long time. And this is for a variety of reasons. It does improve resolution. It also reduces artifacts, such as charging of the sample, and it means you can potentially image samples without coating. Now, these are some electron micrographs I produced many years ago, and showing the deperiodic banding of collagen fibrils in an SEM. And you can see some of the bands there. You can also see the coating that I used because of the resolution of the microscope here. And this problem of having large volume signal and being able to resolve small features has been acknowledged for quite a long time. So this was produced in 2000, this paper, and it was looking at the spatial resolution using EDS. And you can see here with 7 kV, you've got much better differentiation between the layers than you do have at the 20 kV. You've got a lot of blending here. The gold layer here looks like it's inside the copper. And you've got the aluminium layer kind of spilling out into the carbon. Whereas if you drop that voltage, you can get much better resolution. And this is quite a long time ago. This is with some liquid nitrogen cooled detectors, I believe. But there are problems just reducing the electron accelerating voltage. If you do so here we've got a spectrum, it's just on an iron sample at 20 kV, and you can see really nice peaks, and you've got the K lines here and the lighter lines here. But if we drop down to 15 kV, you can immediately see a reduction in the amount of X-rays, excuse me. And again, once we go down to 10 kV and 5 kV, excuse me, I just need to take a drink. And by the time you get down to 3 kV, even your light or your low energy X-ray line is quite reduced. This means you need a really sensitive detector in order to pick these up and make the best of the signal coming from your sample. And again, this is kind of just comparing it. You can see the different spectra, the different accelerating voltages, and you can also see the number of counts. So these are the number of X-rays received at each of these. So at 20 kV, you're getting tons of X-ray. And then as you go down to 10 kV, you can see the effect on the K series. The L series is still good. But as you drop further, you get much, much lower counts. And this affects the speed of your acquisition and the amount of information in your data. So in order to overcome this, we developed the ultimate extreme detector, and that's what we've got here. And this involved several things. So, for example, we changed the electron trap. So you can see the sensor inside the detector here, and this is what senses the X-rays. And you can see it's much closer to the snout of the detector than in a standard EDS system. That's because we modified the electron trap. The electron trap is there when you've got backscattered electrons coming out, and the electron trap is there to make sure they don't hit your detector, because you want to be detecting the X-rays, not the backscattered electrons. We also changed the shape. So if you've got two circular things, they can't get as close together. So the snout of the ultimate extreme detector is more like this, and it means we can actually get closer to the pole piece. We don't have a window, and that window absorbs some of the light energy X-rays emitted from your sample. So by removing the window, we can detect all the X-rays that are coming in. We've also got a large area sensor in there to detect as many of the X-rays as possible. And the electronics had to be adjusted to make sure that we reduced the amount of electronic noise, and we could make sure that we were picking up every single signal we could. We also did a variety of work in the software to help that. And also we had to build in some safety features to make sure that our windowless detector wouldn't get damaged in the microscope. And ultimately, what does this mean? Well, it means we've got a much smaller front of our detector, we can get much closer. And because we can get closer to the pole piece, it also means that the working distance is much better. So you have an optimized working distance of your sample from the pole piece to get the best amount of X-ray signal. And typically that can be 10 or 15 millimeters away from the pole piece, which isn't great for image resolution. By reducing the front end of the detector, we can get closer and we can bring the sample much closer to the pole piece. So it changes depending on the microscope, but it's around 5 millimeters, and it can be pushed a bit further, which means you get much better image resolution as well as much better signal from your sample. And because we're that much closer, we're capturing much more of the X-rays that are emitted. Now, for life science samples, this means that we get a lot more signal, and we've got in particular nitrogen. So here we've got a comparison, ultimate extreme, and our largest normal or standard EDS detector. It's a bit unfair on the Ultim Max because we're only at 6 kV here. But hopefully you can see this nitrogen peak that I've indicated with the arrow. It's really clear in the Ultim Extreme data and less clear in the Ultim Max 170. And you can see that in the maps. We've got nitrogen coloured in yellow here. And this was only 10 frames, so it's a relatively quick acquisition, hence the data slightly noisy, but we're getting much better signal out of the Ultim Extreme than we did with the 170. And that is a lot because we're collecting more of the X-ray signal and we're collecting a lot more of those light elements. And you can see this with standard materials as well. Here we've got some nanoparticles, and this is with a standard EDS, and this is with the ultimate extreme. So this was acquired within 7 minutes at 1.5 kV with a 3.5 millimeter working distance, so really pushing the resolution there. And what you can see is that the X-ray counts we were receiving with a standard EDS are quite low, but with the ultimate extreme, it's enormous in comparison, which means we can get better mapping data, better accuracy, and you can see the results here in these images. Again, that's due to the geometry inside the microscope. Now, when you're looking at X-ray lines, you need to activate the X-ray line, and you can only activate an X-ray line if you're slightly more than 2.5 times its energy in the main beam electrons. So if your X-ray line is 1 kV, you need at least 2 kV, preferably 2.5 kV in your main beam in order to activate it and get the maximum emission from that particular X-ray line. So it becomes quite challenging when you're dropping down your kV, and it means we can't often use the K lines. The K lines are great because they're really obvious and you get lots of information out of them, but you may have to use one of the lighter or lower energy lines, and you can see here on this periodic table where we've highlighted which of the energy lines you need to use and which you can below 2 kV in this example. So I wanted to move on to some application examples because we all like to see real data. Hopefully that's given you a bit of an overview about the ultimate extreme itself. Please do ask me questions about it later. And I want to start off by showing you some material science. So here we've got a semiconductor device, and this is really nice to show you some of the resolution advantages. So like with a lot of life science samples as well, it's got many different elements within it, but it's got really fine structure. And if you look at 3 kV, we're getting some really nice structure through. So we're getting less than 30 nanometer resolution in our maps, which is pretty good. What we have to do in the software is make sure that when we're looking at some of these light elements, that the peaks aren't overlapping. And that's quite common with the light X-rays or the low energy X-rays, is you get a lot of peak overlap. And it's quite important to have the software to be able to differentiate that. And we do, and it's called TrueMap, and you can see the difference here. So here we've got the difference between the silicon and the tungsten maps. And if you remove that peak overlap and you resolve it, it's essentially a deconvolution. You can see the resultant data down here. And you can also see that in the aluminium there, how we're improving the maps and making sure that we're only displaying signals that we're certain that are from the right peaks. So here we have a layered EDS map, and you can see a lot of the resolution and how we've got these nodes that are really well highlighted, and you can see where the oxygen and the nitride structures are within this. And we can go even further, and if we go even further down to 1 kV, we're making absolute sure we're just getting really surface information. And you can see the difference in the electron images here. They're not quite the same region, but they're an analogous region. So you can see the difference. We're getting much more surface information. We're getting much better surface information from both the silicon and the tungsten here. So we're getting much better signal. You can actually see in the tungsten, we're getting much clearer signal as well, because we are just limiting it to that surface information. And this means we can get really accurate data. And here the maps at 1 kV, again, 6 minutes, so fairly quick acquisition. So you're able to then improve surface sensitivity. Here we've got the oxygen maps as a comparison. So we've got the 3 kV where we're getting information from slightly beneath the surface as well, and 1 kV where you can see we've really defined that surface structure. And here are the layered maps showing the same thing. And I think this is quite striking how much more surface information we actually get by dropping it down to 1 kV. And we're actually achieving around 10 nanometer spatial resolution. And here's a different one. This is a phone processor. And again, you can see the really nice differentiation between layers at very low kVs. So again, this is 1 kV, and we're seeing these really nice differentiation between the layers that we just would not be able to see at higher kVs. So now I'm going to talk to you a bit about life science, so my favoured area. So I use the detector for multiple different applications. I don't always use it at the lowest kV because what I like to use it for is that light element detection, which is so important in life science samples, and getting particularly nitrogen signals through makes such a difference to the interpretation of data. And this is an example I was working on with a collaborator. And here we've got an hydroxyapatite implant. And this is a biodegradable implant. We get cells and tissue invading the implant material and laying down new bone material over the course of time. So this is after 1 month of the implant being in place. We've got some bone tissue up here, and this is the implant. We've clearly got something which in the backscatter data is very intense. And this is actually a montage of 240 images. So these have all been acquired at much higher magnification, and we've montaged and stitched them together. Now, it's very difficult in this, even though we've applied some stains. So we applied uranyl acetate as a stain on this sample, and this sample is embedded in resin, and it's been cut and then polished. And you can see some of the marks from the polishing on here. But it was actually quite difficult to really see where tissue and cells had invaded into the sample. Now, this is the nitrogen maps superimposed over the electron data. And immediately you can see where there are clusters of nitrogen within the hydroxyapatite implant. Now, hydroxyapatite does not contain nitrogen. It's calcium and phosphate. So you wouldn't expect to see nitrogen. So everywhere we're seeing nitrogen here is indicative of cellular material. And we can also have a look at the uranium signal. Uranyl acetate targets DNA and various other biomolecules. And you can see that the uranyl acetate signal also overlaps greatly with the nitrogen signal. Where it doesn't, it's likely that we're getting an accumulation of polypeptides that would occur around the edges in the extracellular matrix around the implant. Quite interestingly, we've got a rather strong uranyl acetate signal here. Now, uranyl acetate is also known to bind to collagen particularly well, so it's entirely possible we've got a collagen deposition occurring in these areas, possibly where we've got new bone growth. So we've got lots of different areas within this sample that we can now identify. In here we have the calcium map, and here we have the phosphorus map. And again, you can really see then the difference between the implant and the cellular material within it. If you have a look, we could, because this is a montage, we can zoom in on different areas, and we've got really nice resolution. So this cell in particular is fairly obvious. It's a stellate cell embedded within the matrix of the hydroxyapatite implant. You can see here we've got loads of little processes coming off, and it's fairly obvious even in the electron data alone. But there are other areas around that are not necessarily very obvious. So you can see in the EDS maps these areas where you get the nitrogen and the uranium signals overlapping each other, so you can positively identify that as cellular material. I would be less convinced about identifying that purely on the backscattered data alone. And likewise, when we move over here, even though we've got those stain in the regions, it's really difficult to pick out the biological material. And when you look at the EDS maps and you're able to resolve that, different X-ray signals, you can really see where you've got all these really fine processes. Now this is a biodegradable material, and here we've got the backscattered data. Now again, I wouldn't want to interpret any of this as actually seeing particles of the implant being absorbed or engulfed by cellular material. I would not be able to differentiate that. I'd probably guess here this is biological, but that's about as far as I would get. Now, if you look at the EDS maps, once again, you're immediately seeing some really nice differences, and you're seeing these really small little particles as well that have been either surrounded by or engulfed by the biological material. And this is cellular material here that's working on digesting these hydroxyapatite implants. And we're also able to see areas of the implant where we get a change in the elemental distribution, and this is in calcium and phosphorus. So you've got quite distinct ratios in bone material in the implant, from the 1-month-old implants, and in the 3-month-old implants here, and this is bone, so the same sort of ratios. But if you look here, and this is this area I've highlighted with the arrow, you can clearly see how there's a distinct drop in the phosphorus signals. We're getting much more calcium. Now, we only observed this at 1-month-old. We're not quite sure what's going on here. You can also look at nitrogen content and look at the differences between the bone and the implants. And as mentioned before, you can see there's much less nitrogen in the implants in comparison to the bone, which would be expected, and allows us to differentiate the sample. This is a similar type of situation. Now this is 3 months old. So the implant has been embedded in for 3 months, and the model here was mini pigs. And you can see a difference between the implant and the bone. There is a density difference. There's also a crack that's running along that's due to the sample preparation process. But as before, identifying what actually is going on in the implant, what is biological material that's invaded the implant, what's happening to the biodegradation, it's really difficult to interpret from the backscattered data alone. And if we go to the EDS maps, immediately you can see there's a load of green material in the implant, the kind of peach material, which is the implant itself. A combination of the calcium and phosphorus signals is much more broken up and is in various bits all around the place. And you can see like really big accumulations of nitrogen in here as well. So don't quite know what's going on in those regions. And here you've got the bone. So clearly we can see the difference between the bone and the implant material. And you can see some areas where the implant material actually doesn't follow this crack line. You can see the differentiation much better with the EDS than you could with the backscattered data. So hopefully that's given you a bit of an idea. It's one of the many projects I've been working on where I've used higher kVs. So with the Ultimate Extreme Detector, you can go up to 7 kV, or you can push it slightly higher up to, say, maybe 10 kV. You will get a bit of a background of backscattered electrons hitting it, but it really depends on what you're doing. So these were acquired at the top range of the Ultimate Extreme Detector. So we got a lot of signal, but we had the nitrogen signal in there. And that was the key part of the analysis here. So the Ultimate Extreme Detector is really capable of imaging all sorts of different levels, not just the ultra low kV And once again, this is kind of highlighting the nitrogen difference here that we're detecting. So with the key thing with biological samples is that all of the elements we're interested in are at the lighter end of the table. And everything apart from hydrogen, we can't detect hydrogen with EDS. All of these can be detected, and these are all the elements that would be naturally occurring in samples and considered essential elements for biological material. Now, there still isn't a definitive list of essential elements for any species, which I find quite shocking and surprising. But this is for humans, and these are the relative percentages in human. And you can see that these elements here, they become trace elements. So it's much harder to actually pick them up using a technique such as EDS. It is possible in some areas, and it is possible where you see an enhancement of or an accumulation of these signals. So once you get to below 0.1 weight percentage, it becomes quite challenging. But all the other elements are readily detected, and different tissues and different cells have different levels. This is kind of an estimate over an entire human being. Here we've got an Arabidopsis thaliana leaf. This is an unstained leaf. So I didn't stain them. I embedded them in LR white, which means you've got some density difference. So you've got some electron signal difference between them. And then this was imaged at 2.5 kV for 15 minutes. And we're getting some really nice signals. Again, you can clearly see the nitrogen signals and this really beautiful nitrogen map highlighting those cells. And we're starting to get calcium. We're not really activating calcium particularly well at 2.5 kV, so that's why the signal's a bit patchy, but we are still getting some of the signals through. And one of the areas I've done a lot of work on is with three-dimensional analyses, particularly using serial block face scanning electron microscopy. And this is a growing area of interest, 3D volume, EM, in all sorts of areas, but in biology particularly. And so this particular data set was collected using a Katana microtome. Now, one of the challenges you have with any of the 3D data is the volume of your beam and sample interaction, because you've got a small slice that you're taking off the sample each time. And if you're collecting data deeper than that slice, it's going to influence each layer of data you collect. And with SBF, SEM in particular, what you have is a diamond knife that slices your resin embedded sample, and you image the sample after each slice. Now, what can happen upon exposure to the electron beam is the resin softens slightly. So when you're trying to cut with your diamond knife, it can cause distortion in the surface because it's softened. A way to get around that is to have really low kV when you're doing it. And it becomes a bit of a challenge actually doing that and getting enough signal out of your sample. So often the samples are stained quite extensively. And in this case, this is some lamprey skin, and it had lots of osmium stain in it. What we wanted to do was see if we could use this with the ultimate extreme detector. It would not be possible to use this system with a normal EDS detector because you wouldn't have the right working distance between your sample and the pole piece, and you have the potential for the X-ray signal to be shielded because of the knife position. But with the ultimate extreme detector, because we can get so close to the sample, we can still get a decent signal even at those lower kVs. So this was collected at 3 kV, and we collected almost 200 slices overnight. This was just 2 frames, so we didn't have very long. If you expose the resin block to multiple sweeps of the beam, it softens the resin in the same way, so you get a distortion of your ultrastructure. So we only exposed it to 3 sweeps of the beam. 1 sweep was collecting the backscatter data, and the subsequent 2 frames were the EDS maps. So they're quite noisy, but hopefully you'll be able to see that we were able to get the data, and then we're able to model all of these highly stained, I think they were melanosomes within the skin, really nicely with the osmium, and we're able to model it in 3 dimensions. So my voice really is giving up now, but I just want to sort of finally say that the other thing I've been able to do with the Ultimate Extreme is use low KVs, use uncoated samples, and be able to correlate using atomic force microscopy. And this was a tooth sample. So what we wanted to look at was the exposure of lactic acid and its effects on dentine and enamel in a tooth. So we wanted to see how it affected the topography and also the dissipation, which is one of the measures you can get in an atomic force microscope. So basically the deformability of the material and how that was affected by the chemistry as well. So this meant we couldn't look at a coated sample because it would interfere with the AFM measurements. And we did the EDS first, we took it to an AFM, we did the AFM measurements while it was being digested with acid, and then took it back to the EM. So there's lots of back and forth. This is during lockdowns, involved shipping samples around the country. But we're able to get some really nice data and distributions. And here you can see in the gold colours, this is the dentine, and in the blue, this is the enamel layers. And you've got the before and after next to each other. So you can see there's been quite a big change, not so much in the oxygen, but in the carbon, phosphorus, calcium, and in the nitrogen signals as well. And this correlated with some of the dissipation changes we were observing. So I'm going to wrap up there before my voice gives up completely and open it up to questions. But hopefully I've been able to show you a little bit about why we developed the Ultimate Extreme Detector and the type of questions it can answer, and also how we're able to use it to look at samples at low KVs and get the ultimate resolution out of our samples, but also how we're able to apply it to biological tissue and maybe not aim for that resolution, but look at more the light element emission that's coming from the sample. And I want to thank you very much for your attention and open it up to any questions. And I apologise once again for my voice.
Oh, it's been fine. No, thank you for that. It was a great talk as usual. We do have a couple of questions in the chat so far. So the first one is, can the extreme EDS detector be used with stage bias or beam deceleration modes?
Yes, so my understanding is it can be used with beam deceleration. I'm not sure about stage bias. I would have to check on that.
And they're kind of similar things, aren't they? The beam deceleration stage.
Yeah, I don't see that it would be affected by it. I'm pretty sure you can use it.
Yeah, that's what I thought. And the second question is, what is the energy resolution in the range below 1 kV?
Yeah, that's a good question. I don't know that at the top of my head. It's on our website and our material, but I don't know that. I'm a biologist. I don't tend to think about energy ranges with with it. So I'd have to look it up for you. Sorry, I can't tell you off the top of my head there.
I got one. What's the smallest thing you've been able to analyze with the extreme?
The smallest thing were 10 nanometer gold labels in some heart tissue. So it's challenging because when you've got such a small sample, you get such a tiny X-ray emission that picking that out of the rest of the emission can be really challenging. We did this actually using slightly higher KVs and using a stem holder in an SEM. So what we were doing is we were improving the image resolution and the resolution of the signal by having a very thin sample. and activating as much of the X-rays as possible by having a slightly higher KV. And whenever you've got a sample that small, you do have to use high magnifications. It's the same as with an electron signal as well. You're not going to see the 10 nanometer gold particles unless you're at a magnification where you can actually see that in your pixel resolution. And it's similar with EDS. So you do actually have to go fairly high up in resolution in order to do it. But 10 nanometer gold particles is the smallest thing I've been able to visualize.
It's not there. And I've got another question. Can the extreme be used with fib? Is there like a retractable?
Yes.
Yeah.
Yeah. So we've got it via. So what happens is the fib is controlled by the SEM, and it calls upon Aztec and the EDS detectors to capture images at certain points. The extreme can't be in place and on, or rather active, while the fib beam is milling, because you'll just deposit all sorts of stuff on your detector. So it retracts. The electron microscope calls on it to retract. It retracts. It stays cold, but it retracts behind a flap which protects it. Yeah. So then you can do the fib milling, and it inserts again to capture your data.
Yeah, that'd be awesome. Have a try with that kind of stuff.
Yeah, it's quite cool. I'm impressed.
So has anyone else got any other questions that they'd like to ask? You got a question, Aaron? Are you all right?
I think I've probably asked most of my questions, but I will have, in a few months' time, I will have a lot more questions once our detector's up and running. So yeah, I will be probably emailing on a regular occasion, but there we go.
Do you know when your detector's arriving and being installed?
Not yet, as far as I know. It's the instruments arrived last week, so they're all sat. The new instruments are sat in crates at the moment, and are being installed in a couple of weeks. So I think the detectors shortly after.
I think it should be about around mid-June, maybe. It should be up and running, I think, around mid-June, I think.
Exciting.
It's exciting.
Yeah. Yeah. So yeah, it should be good.
New toys to play with.
Yeah, lots. Yes. Okay, well, thank you, Louise. And yeah, thank you for getting through that as well. I know you're not feeling well and you're struggling. So